Hello students. Now we are discussing the next topic. It's where are the ATP and NADPH used? It is on the page number 215. So where are the ATP and NADPH molecules used, as well as we will discuss the Kelvin cycle today in this video. Students, where are the ATP and NADPH used? So during the light reaction, cyclic photophosphorylation ATP formation take place, non-cyclic photophosphorylation ATP and NADP molecule formation take place. Then chemosynthesis process also ATP and NADPH molecule is formed. So during the light reaction, the ATP and NADPH molecules are formed. These molecules are now utilized for the synthesis of food, and this process we used to say biosynthesis phase. By this process, the glucose molecule is formed by the photosynthesis. So biosynthesis is a phase in which the molecule of glucose is formed in photosynthesis by utilizing the ATP and NADPH molecules. The source of ATP and NADPH molecule is light reaction. So this phase is called biosynthetic phase. This biosynthetic phase is also known as dark reaction, because this biosynthetic phase is not depend on the light. So light is not required. But in this process, the product which are formed in the lights are utilized. So biosynthetic phase glucose is formed, and this glucose formation processes occur in dark. So it is also called dark reaction. So dark reaction are light reaction, dark reaction are biosynthetic reaction. The M. Kelvin and his co-worker, they performed the many experiments on the green alga while taking the carbon 14 radioactive, and they came on the conclusion that the CO2 fixation take place in photosynthesis. It reduced to form the glucose. So CO2 is fixed, and this CO2 molecule form the glucose. So this process is called biosynthetic phase. When CO2 is fixed, then primary product is formed. First stable compound is formed. And if first stable compound is formed, that is 3 carbon atom compound, then it is called C3 pathway. Why it is called C3 pathway? CO2 is fixed, and after fixing the CO2, primary stable product is formed. It is 3 carbon atom product, so it is called C3 cycle. So phosphoglyceric acid is the first primary stable product formed by the fixation of CO2. So CO2 is fixed, and primary product is formed phosphoglyceric acid, and this phosphoglyceric acid is 3 carbon atom compound, so it is called C3 pathway. And such plant where C3 pathway occur, they are called C3 plant. Second, there are the some plants also where CO2 fix, and it first product is formed 4 carbon atom compound. If CO2 is fixed and the first product is formed, that is 4 carbon atom compound, the pathway is called C4 pathway. C4 cycle or C4 pathway. And C4 carbon compound, C4 carbon compound oxaloacetic acid. When CO2 fixed, then oxaloacetic acid is formed, and this first primary product in some plant. So it is called C4 pathway, and primary product of C4 pathway is oxaloacetic acid. So we are discussing first one, C3 pathway. As I told you, C3 pathway, the first primary product is formed. This is 3 carbon atom compound. So Kelvin and his co-worker together they work, and they came on the conclusion that the CO2 reduced to form the glucose, and this C3 cycle occur in all the photosynthetic plant, and this photosynthetic plant we are discussing now. There are some phases in the C3 cycle. So students, Here you can see on the slide the product of Lye reaction ATP and NADP. And this ATP and NADP are used for the synthesis of food. So this process is called the biosynthetic phase, or this phase occur in dark, so it is called the dark reaction. In this dark phase do not depend on the light, but the product which are formed during the lights are used in this biosynthetic phase. Second important part is the dark reaction. Where does it occur? So dark reaction it's occur in the stroma part of the chloroplast. So you can see the reaction involve the biosynthetic phase or dark reaction occur in the stroma part of the chloroplast. Second thing I told to you, if CO2 is fixed and 3 carbon atom compound is formed, this is PGA. And this cycle is called the C3 cycle, or it is called C3 pathway. And some plant rice, wheat, okay, so rose, mango, rice, wheat, whatever plants you are watching in the cool and wet climate, they are C3 plant. Rice and wheat, here examples are mentioned. Other cases, CO2 when fixed up, then 4 carbon atom compound is formed. It is oxaloacetic acid, and such pathway is called C4 pathway. Some plants which are C4 plant called maize, sugarcane, and this type of plants grow in hot and sunny climate. So maize and sugarcane are C4 plant because they fix the CO2, and first primary product is stable product is from this is oxaloacetic acid, and it is 4 carbon atom compound, so it is called the C4 plant. Another Kelvin cycle which is also called C3 cycle, because as I told you first carbon atom compound is formed, that is the 3 carbon atom compound. So you see in the Kelvin cycle stages of the Kelvin cycle we are discussing. First one, it is given 6x of CO2. 6x of CO2, it means 6 molecule of CO2. Then you will see 6x ribulose biphosphate. It is mentioned RUBP. So 6 molecule of RUBP. So student, first stage in the Kelvin cycle is when RUBP is combining with the CO2. RUBP is combining with the CO2 in the presence of RUBP carboxylase enzyme. Then first unstable compound is formed, and then it's from the stable compound, it is 3-phosphoglyceric acid. So 3-phosphoglyceric acid, it is the phosphoglyceric acid, and it is 3 carbon atom compound. So this is the first stage where RUBP combined with the CO2 in the presence of RUBP carboxylase, and 3 carbon atom stable compound is formed, it is phosphoglyceric acid. This is called the carboxylation process. So first stage is carboxylation. Then second stage, the phosphoglyceric acid, it reduced by using the ATP 12 molecule and NADPH2 12 molecule. So first phosphoglyceric acid change into 1,3-diphosphoglyceric acid. Then using the NADPH2 12 molecule, it convert into 3-phosphoglyceraldehyde. Then it convert to 2-glyceraldehyde phosphate, and ultimately it form the 1 molecule of the glucose. So this process where CO2 reduced to form the glucose, it is called the reduction process. So in this stage, a series of reaction occur. In this state involve the utilization of 2 molecule of ATP for the phosphorylation. Each CO2, it means 6 CO2 are there, so it is 12 ATP molecule. Similarly, each CO2 requires 2 molecule of NADPH for the reduction of CO2. So there are 6 CO2s, it means total 12 NADPH. So overall in the reduction 12 ATP and 12 NADPH2 molecules are used. So the fixation of 6 molecule of CO2, this type of 6 turn of the Kelvin cycles are required. So this process where phosphoglyceric acid is reduced by using the 12 ATP and NADPH2, and then ultimately it forms the 1 molecule of the glucose, it is second process which we use to say reduction. First was the carboxylation and second reduction. And then what happened, the phosphoglyceraldehyde, then it is changed to the glyceraldehyde, then ribulose monophosphate, and then ultimately RUBP is formed. So this process where the regeneration of the RUBP occurs, and regeneration of the RUBP, it means the molecules of RUBP is formed. So the regeneration step require one ATP for the phosphorylation of one. So one RUBP molecule is formed, then one ATP is required. So here six RUBP molecules are formed. So six molecules of ATP required. So this process where the RUBP is formed, it is called RUBP. It is called regeneration. So in Kelvin cycle there are the three steps only. One carboxylation, another is reduction, and third is regeneration. Now this same process I am explaining in these phases also. In our book this diagram is given. So RUBP 1,5 biphosphate, it is RUBP only, combined with the CO2, it fixed the CO2 from environment, and that in the presence of RUBP carboxylase, that converted 3-phosphoglycerate. So this is the first step of the C3 cycle. It is called carboxylation. Then 3-phosphoglycerate reduced by using the 12 ATP and 12 NADPH, and ultimately what happened? One molecule of the glucose is formed. Later glucose convert to sucrose and starch. So this process is called the reduction process. So second process is reduction. Third is the triphosphate, again it form the RUBP molecule. So here formation of RUBP molecule, again 6 ATP molecules are used to form the 6 molecule of RUBP. So this third process is called the regeneration process. So Kelvin cycle we have three process: carboxylation, reduction and regeneration. In this one detail you can see CO2 acceptor molecule is RUBP. In Kelvin cycle the CO2 is accepted by or fixed by RUBP. So this pathway RUBP is the acceptor of CO2, and in this pathway the main product is formed. What is the main product? Glucose. In the pathway glucose is formed, so it is called Kelvin cycle on the name of the M. Kelvin. And this discoverer of this pathway, Kelvin cycle stages 3 I have told you, and this to see summary of the carboxylation. What is first step of the C3 cycle? Here is the fixation of CO2 into stable organic compound. CO2 is fixed and PGA is formed. CO2 combined with RUBP phosphate to form the PGA. So this process occurs in the presence of RUBP carboxylase enzyme. This is carboxylation. Second, as I told you, there are series of reaction occurs through which what happen? This phosphoglyceric acid is converted into a molecule of the glucose. So 2 ATP, 2 molecular ATP for the phosphorylation, and 2 molecule of NADP for the reduction of the each CO2 molecule. So in reduction total 12 ATP molecule and 12 NADPH2 molecules are used. So in this process what happens? 6 molecule of CO2 for fixation of 6 molecule of CO2, 6 turn of Kelvin cycle is required to synthesize the one molecule of glucose. And third stage of Kelvin cycle is the regeneration. Regeneration of CO2 acceptor molecule RUBP. So again the molecule of RUBP is formed. Earlier the molecules of RUBP is fixing the CO2. Now what happened? Again this RUBP molecule is formed, and in this to continue uninterpreted. So for if this process to be continued, regeneration step require 1 ATP of the phosphorylation. So 6 molecule of RUBP are formed. So there 6 ATP molecules will be required. Hence for every CO2 molecule entering in the Kelvin cycle, 3 molecule of ATP and 2 molecule of NADP. So every CO2 molecule, it means total 12 ATP, and uh, for every CO2 molecule entering in the Kelvin cycle, 3 molecules of ATP and 2 molecule of NADPH are required. So total of the conclusion of the every molecule of CO2 in the Kelvin cycle, 3 molecules of ATP and 2 molecule of NADPH is required. 3 molecules of AT for every CO2. So there are the 6 molecule of CO2. Okay, so 6 molecule of CO2 for how many? 6 3 ja 18 ATP are required. So in Kelvin cycle, it is the summary in and out. So what are used in Kelvin cycle? 6 CO2 molecule, and what we are getting? 1 glucose molecule. Here we are using, it is required 18 ATP molecule and 18 ADP molecule are released. Here it is used 12 molecular NADPH and 12 NADP molecules are used. So it is a summary of the Kelvin cycle. So the students overall Kelvin cycle is given by M Kelvin. It is called the C3 cycle. In C3 cycle, because this the first primary product is from phosphoglyceric acid, which is 3 carbon atom compound. So it is called C3 cycle. And C3 cycle, carboxylation, reduction and regeneration, these are the three stages. Thank you.